Thank you for kind introduction. So I'm Masahito Hosokawa from Waseda University and bitBiome in Japan. First of all, I'd like to thank the organizers and Oxford Nanopore Technologies for giving me a chance to talk today. So I'd like to talk about the single-cell long-read sequencing of human microbiome to obtain a complete genome uncultivated single cells. Here is the COI to disclosure. So microbial single-cell genome sequencing is culture-independent genome recovery technology that serves as an alternative or synergistic technology for the conventional metagenomics. SAGS stands for the single-cell amplified genome, provided as the strain-resolved genomes, with linking the mobile genetic elements, including phages and plasmids. It is characterized by applicability to the rare microbes and small biomass and highly diverse samples. Single-cell genome sequencing has performed in the two steps shown here. The first is isolation of microbial cells from the sample, and the second step is whole genome amplification and sequencing. However, the single-cell microbial sequencing has faced challenges. So as you know, the bacterial cells has different shapes and sizes, and their membrane structure differ. But we have to handle these ununiform microbes at once from the sample. We had to amplify their tiny DNA million-folds to obtain enough amount of DNA to carry the next generation sequencing. So the whole genome amplification is quite important step. So MDA stands for the multiple displacement amplification. Is a general method for whole genome amplification from the single cell. It uses the Phi29 polymerase and random primer, and MDA provides us a very long fragment with low mutation rate. But the using of random primer, we have to prevent the contamination from the sample source or laboratory environment to get the precisely amplified bacterial genomes. So to fight with the contamination, we had to prepare the special equipment, including the contamination-free environment as shown here, or some equipment for massively parallel reaction for single-cell genome amplification reactions. So we also need the highly trained operators as well, but as you can imagine, so it is not easy to complete this setup. So we have been developing a new technology that make the single-cell microbial genomics more easier and more effective and more data productive. Here is our solution. Our solution is the application of microfluidic droplets for the single-cell reactions. So microfluidic droplets is a great platform because it can reduce the risk of contamination by reducing the reaction scale from the conventional microliter scale to the picoliter scale as a droplet. The workflow is straightforward, starting from the encapsulation of microbial cell inside the droplet, and all of the lysis and whole genome amplification processes are conducted in the compartmented droplets in a single tube. Based on these concepts, we have developed the technology named SAGGel, single-cell amplified genome in gel. As shown in this video, we can encapsulate microbial single cell into the droplet. And these droplets contain the agarose. So we can solidify the droplet to form the gel base. So gel matrix support the cellular structure and DNA without escaping the DNA outside the droplet. So by simply immersing these gel base in the various solution, we can process the cell ratios and the DNA amplification inside the gel base. After the genome amplification, we can collect the gel base containing a single cell amplified genome one by one by using a cell sorter. So we can preserve these amplicons for the barcoding sequencing. So SAGGel, now commercialized as bitMap, provides us the high quality SAGs of human gut bacteria in faces. This slide shows the comparison of our SAGGel technologies with the conventional method. It uses the direct cell sorting and plate-based reactions. So these dots represent the 96 SAGs sorted from the highest genome completeness, the lowest genome completeness. SAG0 showed the average of 75% genome completeness. On the other hand, the conventional one showed 14% in average. So then we applied our technology to the over 50 healthy volunteers and patients, and we obtained 20,000 SAGs from human microbiome, including the feces and saliva sample. This is a cladogram. It shows the phylogeny of SAGs we recovered, and different colors show the different sample origin, the feces and saliva, and the blue show the genera found in both of feces and saliva. So as you can see, the SAG can be recovered from the variety of taxonomy, and it includes the typical human microbiomes. And some genera commonly obtained from both of feces and saliva. We also do the comparison of our SAG with the metagenome assembled genome in the conventional metagenomic binning strategy. And this cladogram shows the MAG and SAG in a different color, and both blue show the genera found in both MAG and SAG. From our initial evaluation, some of the taxonomy has recovered only from the MAG or SAG. These biases may reflect the differences in gram-negative and positive. So because the single cell genomics requires cell integrity, it requires DNA maintained inside the cells. So here, I'd like to show you some point in which SAG resolve the problems in the short-read MAG. The first is ribosomal RNA gene recovery. In the short-read MAG, highly conserved sequences, such as the ribosomal RNA gene, often most lost in MAG, but the SAG can recover in the cell-by-cell basis, so you can recover the ribosomal RNA with higher recovery rate compared to conventional MAGs. The second is to elucidate the association of phage and host. SAGs can recover each strain, so it is possible to determine which strain has which gene, which mobile genetic elements. And also we can analyze the location of genomic area. The third is the target selectability. So if you have some interest in the sample, we can get viral bacterial specific single cell sequences or the bacteria with target gene sequences. So from the diverse sample, we can correct the specific bacterial genome with using this single cell approach. Because the time is limited, I will skip the detail of this technology, but you can get the disadvantage by using the single cell analysis approach. So today, I like to focus on the long-read single cell sequencing. So because the single cell short-read SAG still faces technical challenges, that is the genome fragmentation. Here is a table. It shows the bacterial SAGs obtained from short-read SAG from the single figure samples. So as you can see, we could obtain many bacteroid species or strain level genomes, and these genomes show high enough completeness with low contamination that the number of contig ranged 100 to 500. So it means highly fragmented. Ideally, we should make these contig into single circular genomes. So we tried long-read sequencing using the Oxford Nanopore Technologies bitBiome. So the first trial is for the E. coli, culture E. coli SAG with standard assemblers. So with the commonly used assembler as is, we can reduce the contig to 2 orders of magnitudes, but the genome completeness is quite partial, so it means quite incomplete genomes. So the reason why is the biases in the bias in single cell amplified genome. A red area shown here prompt missing from the contig because due to the low read depth. So we should handle these biases. So we decided to develop the new long read assembly pipeline. So we named that Scholar. So it aims to recover low frequency sequences into contigs by debiasing the sequencing depth. The scheme is very simple. Identifying the overrepresenting sequencing regions by mapping the low reads to assemble the contigs. After that, we normalize the low reads as a debiased reads and then reassemble to contigs. So this mapping and debiased cycle is repeated until we get the debiased contigs as improved continuity contigs. By repeated cycle, the contig continuity are gradually improved. And the end of the third of the first cycle, we got the 2 contigs as complete genome and F plasmid as of the E. coli K12 strain. Now, this contig quality is superior to the short read SAG and comparable to bulk DNA. Then we applied this pipeline to the real human microbial samples. And we collected 12 human fecal samples. And finally, we got that 16 strains of 3 gut bacterial species, including the A. Anylospermia hydrois and Agatobacter rectoris and Luminococcus guanabas. As shown here, we did the alignment of these circular sacs from the uncultivated human gut bacteria, and that revealed the many presence of the strain-specific genomic regions. By the detailed analysis, we revealed the strain-specific sequence region concentrate the viral like genes, such as a prophage. So these strains uniquely have their own phage-like genes in their genome, and the sequencing access and location highly depends on strains. Then we applied this pangenome analysis of heterogeneous genomes, and we found the structural variation in polysaccharide metabolic pathway. In this slide, I show the laminose degradation gene clusters, and the bottom side is the inositol degradation gene clusters. We got 9 strained single cell circular genome in this study, and it included 2 cohabiting groups, A and B. So as you can see, laminose degradation pathway were only observed in the 3 strains out of 9 and not in others. And the inositol degradation pathway showed some deletion in some strains. So those presence or absence or gene cluster structure highly depends on strains, and highly depends on strains among their cohabiting families. So our results show that these polysaccharide metabolic capacity highly diverse within the species, and strain clonality may strongly related to the human-host relationships. So here's a summary of my talk today. So we provide a unique single-cell genome sequencing platform named SAGGel and commercialized as a bitmap. And we applied our technology to with the long-read sequencing to get the circular genome from the single cells. And it delivers strain-level profiling and insight into microbiomes with the conventional metagenomic analysis. And we have the use cases, not only animal microbiome as introduced today, but also environmental microbiome, such as soil microbiome, plant microbiome, and seawater microbiome. If you have an interest in our technology, please visit Poster 73, or we have also an exhibition. Please visit the second floor. This is the final slide. I'd like to thank all of the members who supported the research. Thank you for your kind attention.
Thank you. Thank you very much for this the very interesting talk. Now it's time for the questions or comment from the floor. Any questions? Quick questions from me. You mentioned about the targeting the bacteria with the viable cell with PMA or targeted gene sequence. Can you identify the single cell with the particular the immunological characters such as the antibodies? You can targeting an epitope. Can you targeting some particular bacteria with your technology, or you can evolve a bit further? Can you comment on that?
Thank you. We've never tried it yet, but we can treat the cells with antibody, and we can make some technology to specific bacterial amplification of your specific target from the antibody reactions.
Okay, great. Okay, thank you very much.